So welcome back to the session on lipids, and we have been discussing on the classification of lipids, where we saw that the lipids are classified into simple, conjugated and derived, and we also discussed in detail the different kinds of simple lipids. Okay. Now regarding the conjugated lipids, or it is otherwise known as compound lipids, the conjugated lipids are the lipids where you can find on hydrolysis fatty acids and alcohols. Plus, apart from these, it may also contain other additional groups such as phosphate, base. I hope you remember what is a nitrogenous base we are referring here. Okay, a carbohydrate or a protein. So, simple lipids on hydrolysis gives only fatty acids and alcohol, while compound lipids on hydrolysis it gives fatty acids, alcohol and some other compound like phosphate, carbohydrate, protein, etc. Okay. So based upon the compound, additional compound, we name the compound conjugated lipids. Okay. So simply, it is actually esters of fatty acids containing groups other than alcohol and fatty acid. Okay. So it could be phospholipid wherein they have fatty acid, alcohol plus a phosphate group. Okay. Glycolipid where it is fatty acid, alcohol plus carbohydrate. Okay, so likewise we have different kinds: phospholipid, glycolipid, sulfolipid, lipoproteins, etc. So we will be learning in detail more about phospholipids. Okay, so what are phospholipids? So as already mentioned, phospholipids are they contain phosphoric acid residue, that is H3PO4. Okay, it is H3PO4 residue in addition to fatty acids and alcohol. Okay, they are also referred, phospholipids are otherwise referred as phosphatids. Okay, they are known as phosphatids. They are widely distributed. It is found in, from bacteria to plants and animals, and we can see they are derivatives of phosphatidic acid. Okay, phospholipids are derivatives of phosphatidic acid. So, what is phosphatidic acid? Phosphatidic acid, you can see here, this is the alcohol. Okay, this is the glycerol, CH2OH, CH2OH, CH2OH. That is a glycerol, isn't it? Okay, so here what has happened, at each alcohol group, some other molecule, some other compound has got esterified. Okay, so here what is it? It is actually R1COOH. Okay, so that means it is a fatty acid. So here the alcohol is esterified with a fatty acid. Second alcohol on glycerol is also esterified with a fatty acid. The third alcohol, it is actually bound by phosphoric acid. Okay, so it is phosphoric acid molecule, and this compound is known as phosphatidic acid. Clear? It is known as the phosphatidic acid. Now, so this is actually, phospholipids are derivatives of phosphatidic acid, right? And they are amphipathic molecule. What is amphipathic molecule? That is, it contains both a hydrophilic and a hydrophobic end. Okay, hydrophilic end is considered to be the head region. Okay, head end. Okay, it is polar because it is hydrophilic, isn't it? Polar hydrophilic head, and on the other hand, the non-polar hydrophobic region is considered to be the tail. Okay, so head or the hydrophilic region end is formed of negatively charged phosphate group. Okay, so you can see here the 1 minute. Okay, so it is actually O minus. Okay, 1, 2. These are negatively charged. Okay, so it is negatively charged. Okay, so negative phosphate group and positive alcohol group. Okay, so the head is formed of, or the hydrophilic head is formed of negatively charged phosphate group and positively charged alcohol. Okay, now what about the tail? It is formed of the long fatty acids. Okay, this is formed of fatty acids. So actually, this forms the head region. You can see here hydrophilic. Okay, so this forms the head region, while this long hydrophobic fatty acids, they form the tail region. Clear? So this is the amphipathic molecule. So in general, we can see the phospholipids contain fatty acids with carbon 16 to 18 and maybe unsaturated fatty acids wherein it may contain like C18 or C20, that is more frequent. Okay, so these are the more frequently found fatty acids in the phospholipids include saturated fatty acids with carbon 16 to 18, 16 or 18, okay, even number. Unsaturated fatty acids with carbon 18 or 20. Okay, there are two major groups of phospholipids. They are phosphoglycerides and sphingomyelins, and this is based upon what kind of alcohol comes into play for the formation of the phospholipid. If it is glycerol, we call it as a phosphoglyceride. Okay, that is, phosphoglyceride contains glycerol as the alcohol, fatty acids, and phosphate. Clear? Sphingomyelins are those phospholipids with what you call sphingenin instead of glycerol. So instead of glycerol as an alcohol, they contain sphingenin, or it is otherwise known as sphingosin. Okay, so sphingomyelin will contain sphingosin as the alcohol, fatty acid, and phosphate group. Okay, so these are the two major groups of phospholipids, phosphoglycerides and sphingomyelins. And these two you will be learning it in detail, the structure and the functions. Okay. Now, minor phospholipids, on the other hand, include plasmalogens and cardiolipids. Okay, we will be learning it in detail later. Okay, so here we can see this is the glycerol molecule, CH2OH, CH2OH. Okay, here the first two carbon have been esterified with fatty acids, while the third one it is with phosphate. Okay, and this glycerol molecule plus the phosphate forms the hydrophilic head. You can see here glycerol plus the phosphate, it forms a hydrophilic head region, okay, or polar region, right? While the fatty acid part, it forms the hydrophobic tail. Fine. You can see this one, it shows it is CH2, 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 CH2 till CH3. It could go for C16 or C18, right? And you can see it is a straight chain almost, right? And here this is actually saturated fatty acid. The first one is saturated fatty acid. While this one here, you can find a double bond in between. Okay, and this is actually a unsaturated fatty acid, right? And in unsaturated fatty acid, you can find that there is one or more double bonds, and wherever there is a double bond, there is a kink, there is a bend. Okay, so this represents this is just what you call, you may have seen this ball and stick model, isn't it? So here you can see there is a bend. Okay, what happens here? There is a space between, right? If it was a saturated fatty acid, what will happen? It would have gone straight, isn't it? Almost parallel to the first one. So there won't be any space between. It will be closely packed, right? But when there was a bend because of the presence of double bond, what happened is there is a increase in space between the fatty acid. Okay, so you can see that to increase the space, you can actually have unsaturated fatty acid as one of the fatty acids in the phospholipids. Okay, I hope it is clear. Okay, so hydrophobic tail region here and hydrophilic head region here. Clear? Okay, so this is the part of the phospholipid which can bind with water molecules or any other polar molecules to hydrogen bond, while this hydrophobic tail usually remains hidden within. Fine. So we saw that phosphatidic acid, it forms the what you call base for the formation of phospholipids. They are actually the these give rise to the phospholipids, isn't it? Now we also mentioned phosphoglycerides. These are the phosphoglycerides being the major group of one of the major group of phospholipids. Okay, phosphoglycerides contains two fatty acids attached in ester linkage to the first and the second carbon of glycerol, and a highly polar or charged group attached to a phosphodiester linkage to the third carbon. Okay, so clear? Okay, now based on the attached alcohol, we are speaking about the phosphoglycerides. Okay, based on the attached alcohol, phosphoglycerides are of two types. Okay, they are phosphatidyl choline and phosphatidyl ethanolamine. Okay, phosphatidyl choline, it is actually the phosphoglyceride which have lecithin as the alcohol. Oh, here you can see. Fine. So this is one alcohol, ethanolamine. You can see alcohol. It is here. Okay, this is yet another alcohol. You can see OH group. This is choline, right? So if this one was So instead of this whole structure, if we had only OH, what is this compound? This is the compound. Okay, it is phosphatidic acid. Okay, it is phosphatidic acid. So phosphatidic acid do have a glycerol molecule, right? Then this is the glycerol part, fine, and this is the fatty acid part, okay, and this is the phosphoric acid part. Okay, if instead of the amino alcohol unit, if we had a OH, we call this as a phosphatidic acid. Okay, so it is actually the phosphatidic acid which give rise to different kinds of phospholipids or phosphoglycerides. Okay, so in one of the phosphoglyceride, what happens is instead of H, okay, instead of OH, okay, instead of OH, what happens? Another alcohol binds here. Okay, it could be ethanolamine. If it is ethanolamine, what happens? Right, it will give rise to a unique kind of phosphoglyceride which is known as the phosphatidyl ethanolamine or cephalin. Clear? Okay, if it was ethanolamine over here, we call this particular phosphoglyceride as cephalin or phosphatidyl ethanolamine. If it was choline, instead of this part, if it was choline, okay, it would be, what is the name? Phosphatidyl choline, or it is otherwise known as lecithin. Is it clear? So depending upon which alcohol, whether it is ethanolamine or choline, we can identify we can actually find two different kinds of phosphoglycerides. Okay, and these are actually phosphatidyl ethanolamine, that is cephalin, and phosphatidyl choline, that is lecithin. Okay, clear? Right. So this is the structure: phosphatidyl ethanolamine and phosphatidyl choline. Clear? Okay. So why it is known as amino alcohol? You can see here amino alcohol, isn't it? Why it is known as? This is amino and here alcohol. Okay. Amino and alcohol. So that is why it is known as a amino alcohol. Okay. Then what about this one? Here, instead of NH3, the hydrogen has been replaced by 3 CH3 molecules, isn't it? In ethanolamine, we have NH3+, isn't it? Instead of that, what has happened? So instead of 3 hydrogen atom bound to nitrogen, it has been replaced by methyl groups. So it is CH3 3 times. Okay. So here again, you have an amino group and a modified amino group and a hydroxyl and alcohol. That is why it is known as amino alcohol. Okay. So it contains amino alcohol, fatty acid, and clear. Okay. So we'll see the detail. When we are speaking about the phosphatidyl ethanolamine, these are simply the ethanolamine derivatives of phosphatidic acid. I hope it is clear, right? They form zwitterions. Okay. What are zwitterions? Zwitterions are where we can find both positive and negative ends on the same molecule, right? So we can find positive charge as well as negative charge on the same molecule. If we can have such a kind of a compound, we call it as a zwitterion. Okay, this dipolar like ionic compound. Okay. So we can see that the cephalins, they are ethanolamine derivatives of phosphatidic acid, and they are zwitterionic. What was the other name? It's known as cephalin. Okay, cephalin. Okay, why it is known as a cephalin? Because it is found abundant in brain. Okay, that is why the name has come cephalin. Okay, and cephalins, they are found in the brain tissue and nerves and play a very important role in blood clotting as well. They are also found in the cell membranes, protoplasm, etc. Okay, so phosphatidyl ethanolamine. It is ethanol. Okay, it is A over here and O over here. Okay, ethanolamine. Fine. Now next one.